All right. So this first slide is getting into just some of the stuff I already talked to you about. Those are the textbook and the lab manual covers in case you're looking for it at the bookstore. And that repetitive studying idea. So if you are to study every day a little bit and don't study for long stretches of time, like 20 minutes to a half hour, where you're really studying the content, not just letting it bounce off your eyes, but actually studying the content, where you really engage with the material. For me, I couldn't do it in the library, even though I'm sure it's a beautiful library. Although now they probably let you talk and walk around. Back when I studied, I get yelled at if you talked. But anyways, I couldn't just sit and read my notes. I would fall asleep. So I had to get up and walk around. So I usually studied in my room where I could walk around, I could talk out loud, I could use things in my room as visual anchors to what I was learning. And so every brain is a little different on how you like to learn, but you have to engage with the material. Just repetitively reading something over and over doesn't guarantee you're going to know it on exam day. All right. So that's one of the downsides of flashcards. Flashcards can be useful, but if you overuse them or use them in the wrong way, you delude yourself that you understand the material. And so illusions of mastery is the phrase they use in education. They say it's illusions of mastery. You think you know it. Have you ever done that? Oh, I know this. And then you go and take the exam. You're like, wow, I didn't know that. I did really bad on that. And so illusions of mastery, be careful about that. Repetitive studying does work because it lays down new neural pathways in your brain. You're actually physically changing your brain and solidifying those neural pathways so that you can retain the information. What you're trying to do is get this information from short-term into long-term memory. And you need it into long-term in order to recall it on exam day. If you have things just in short-term memory, which happens when you cram for an exam, it's just so superficial. And if you get stressed out, I don't know about you, but when I used to short-term study, when I had it just in my short-term memory, if I got stressed out, it would just kind of bubble away from me and I would just draw blanks all the time when I was trying to answer a question. So just be careful about how you study. Try to find a method that works for you. I'm happy to talk to any of you more about that because I firmly think that if you find the right method for you, your stress level is going to go way down on exam day, plus just in general. I had so much fun in college once I learned how to study. Before that, I plodded along. But once I learned how to study, man, what a difference that was for me. So please give that some serious thought. And you do need good time management if you're going to do good study strategies. Yeah, so that's always a struggle, but we try our best and lecture attendance, lab attendance, and then please read the assigned sections of the chapters. Now, when you think about microbiology, you can take it in so many different directions. So this is showing you the idea of what you can do with some microbiology training. And I'm trying to emphasize that once you know some microbiology, all these other fields of study you can apply that to. I can't tell you how often I've had employers or former students come back and say, yeah, the reason I got this job is they saw on my resume I had micro training. So between what you're learning in lecture and what you're getting in lab, this can make you very marketable. So keep that in mind. And lots of options to take this. I ended up taking it into the plant world because I was very interested in helping plants fight off pathogens and survive and become more nutritious for us to eat. And then here you can see there's a lot of different areas of microbiology. This isn't even fully inclusive. There's other options, but there's a lot of ways you can take microbiology and run with it. Some fun facts. Microbes rule. Probably put that on my tombstone. Microbes rule. They outnumber all other species and make up most living matter, believe it or not. You often hear quotes about other organisms that have this claim to fame, but they always forget about the microbes. Always. Microbes get short shrift on this. They rule. We are about equal. When you count up all of your cells in your body, you're about 1.3 bacteria to one your human cell. So you are more microbe than you are eukaryotic, which is flippy. The first time I learned that, I'm like, what? So when sperm and egg united and it became you, and you grew to 9, 10, 12 months, I guess, for pregnancy, right? And then you were born. Even when you're in utero, you're getting colonized by microbes. Although that is somewhat controversial, but there's more evidence coming out about that. They know pathogens can get in, but they were wondering about the good ones. And now they're saying yes, the good ones are coming into the amniotic fluid and the tissue of the placenta. Anyways, by the time you're born, you're getting to be colonized. And then by the time you're an adult, it's a 1.3 to 1 ratio. So you have more microbe cells in you than eukaryotic cells that came from that sperm and egg uniting and developing. Pretty cool. So they really drive the chemistry of life on Earth and definitely affect global climate. Absolutely. When way back when, decades and decades ago, when they were trying to figure out, how do we figure out if there's life on Earth? You know who they went to? A microbiologist, because she said, this is Dr. Lynn Margulis, she said, if you don't even have to land on the planet. You can just take air samples in the atmosphere of that planet, and I can tell you if there's microbes there with good chance that she's right. And so they definitely drive global climate. They changed the atmosphere of Earth as it was evolving. And they were here first on Earth. Microbes were here first, long before we showed up. And we're going to learn they can be super extreme. They can exist in environments that would kill us instantly. They not only exist in some of these environments, but they actually thrive. What I mean by that is they've adapted to it. They're carrying out their life. They're reproducing themselves. So it's like their niche. They're like, yeah, I can do this. Microbes are fun. How about some bioluminescent furniture? I'm waiting for that to hit the market. I haven't seen it yet, but there was a group in Australia that was working on bioluminescent, like a recliner. So here this guy is reclining and it's all bioluminescent glow. I think that'd be pretty fun. You can buy bioluminescent orbs that you can put on your table. You can buy these units to put in your kitchen where you can grow your own microbes and eat them in a fruit smoothie or something like that. So this one is growing spirulina, which is actually a cyanobacteria. Although if you go to their website, I think they call it an algae, which drove me nuts because it's not. It's a cyanobacteria. So they need a microbiologist on their staff to get that correct. And they're looking more and more at algae more than ever now. Algae is a hot topic. We don't cover that a lot in this class. I do in BI 230, but they're looking at this idea of functional art, where you have something very artistic like lamps, but they're actually growing algae for food for you. And so you may see something like this in the not too distant future, which would be pretty cool. And I say this to all my non-biology friends, most microbes do not cause disease. Now, I know if you took Med Micro, you got a lot of info about disease and pathogenesis, rightly so. However, that's a very narrow slice of all the microbes that are out there. It's a minuscule percentage that actually causes problems. The vast majority either don't care about us or they're helping us. All right. And we'll learn a lot about the ones that are helping us later in the semester. They're using bacteria as tractor beams. So I liked how that was the title of that. So they're actually using microbes to bring radiation to cancer tumors. So they're being used as tools in the health realm. So there's all kinds of examples of how microbes are getting used in these types of ways, which I hope to talk to you more about as the semester progresses. They're even used to refresh and clean off art. And so here, Michelangelo, the famous sculptor, they actually applied painted bacteria onto these sculptures over in Italy because the sculptures were so grimy and dirty from centuries of not being cleaned, and they didn't know how to clean it. They were so worried about destroying it with using, cleansers. So they're like, how do we do this? And so, it gets grimier and grimier. And then a microbiologist said, hey, why don't we paint some microbes on it and have the microbes eat off all the grime? Because microbes will eat the weirdest stuff. And for them, it's great food. It gets them their nutrition, their ATP. They're happy as clams. And in the middle of all that, they get to clean up the artwork. So this was just a short blurb where they were, where's the, here. So they used a couple of different bacteria. We'll be working with Serratia in class, in lab, and then they use Pseudomonas, Rhodococcus, Xanthomonas. So these are all well-known bacteria that like to eat really weird toxic stuff that would be toxic to us, and yet they use it as food and their source of getting ATP. So they actually did these experiments where they went to a obscure part of the marble and were painting on different microbes and looking to see which microbes would clean up the grime. And they hit on successful mixtures. So now they've been able to restore these sculptures without any damage at all. So pretty cool. Speaking of art, microbes are beautiful. Those are microbes growing on plates, agar plates. You'll get to grow microbes on agar plates a lot in this class. So they color coded it on this one so you could see better. But basically, often what you do here is you provide an agar recipe that's very low in nutrients, and then you spot some microbes onto the plate. Like this one was probably spotted here, and then they incubated the plate. And what happens is the microbes are like, geez, there's not much nutrition right here. I better move and find some. So they go forage, and so they start growing across the plate into these beautiful designs, depending on the species. So you can create some really cool artwork. And then this one was done on purpose, where they painted the plate, so to speak. They dabbed different colored microbes onto the plate, and then they created our artistic design. I think you can see it's the egg and sperm idea here, where the one sperm has won, all the others lost out. Here's another one. So that is just someone painting with microbes, with a solution of microbes, and then incubating the plate to get it to grow into these designs. Pretty phenomenal. This is a contest every year that they do. It's called Microbe Art, I think. And some of the people who enter their pieces are amazing. So that's all just microbes. And, microbes can be different pigments. So they pick the ones that are pigmented, these different colors. This one was someone really liked jazz, and so they've created a bunch of plates that depict jazz. And then that, again, that's all microbes across the plate growing. And that one too, beautiful. So this one was really interesting because they put two microbes down, one that is the green color, and then a second one that wasn't, is just clear. And when they studied the plate more carefully, they realized that the one that was clear was non-modal. The other one, the green one, was modal, so it could easily move out across the plate. But the other one was non-modal, but what it did was it would hitch a ride with the modal one. It would like attach to the surface of the modal bacteria and kind of, they call it like surfing, riding the wave of the modal bacteria. They still don't know exactly how that bacteria is doing that, but basically pretty ingenious idea there for the microbe to say, well, I can't be modal, but I'm going to hitch a ride on the neighbor who is modal. And they were able to colonize across the whole plate. So pretty fascinating stuff. Get it? It can't get pasteurized, or because it gets pasteurized. By the way, pasteurization does not sterilize your milk. If you drink dairy milk, there's microbes in it. Good chance of it. When they pasteurize, they just get rid of the nasty ones. They know that those pasteurization procedures will kill things like Mycobacterium tuberculosis and things like that. But that does not guarantee, unless it says ultra sterilized or something like that, normal milk can have microbes in it. That's why it goes bad so fast. So, all right. That's also a microbe artwork, the beer with the foam on it. All right, so I start with microbial evolution because in biology, nothing else makes sense except in the light of evolution, which is so true. So if we can understand and kind of look at how microbes do their thing through the lens of evolution, it becomes more easy to grasp why they're doing certain things. Because we're going to find out that microbes are just, they can behave in some really weird ways from our perspective. But when you look at their life, it's totally understandable if you look at it through the lens of evolution. And so I need to talk to you a little bit about that. And when we are trying to study microbes, the old microbes that are fossilized and all of that, one of the things that microbiologists will turn to is to go to these structures called stromatolites. And there are ancient fossilized stromatolites that are billions of years old, but there's also modern living stromatolites. So if you're lucky enough to go to Australia, perhaps, and along the coral reef shoreline, you will see these. You can actually walk amongst these. And like these are probably up to shin high. And you can at low tide just walk around and look at these stromatolites. And what they are layers and layers and layers of bacteria that have formed these mats that just keep growing on top of each other over the years. So the picture on the left is showing a little bit better how that layer pattern is looking. And you can see the fossilized ones are actually, have sedimented and they have calcium, so they fossilize. And some of those are over 3.4 billion years old. So here's communities of microbes that have been hanging out, we're living back in that time period. Some more recent evidence is they're speculating that microbes existed closer to 4 billion years ago. So it's really hard to kind of wrap your head around that. 4 billion years ago, microbes were here. And we are just a blip in comparison to how long we've been living here on Earth compared to that. So it's hard to find fossilized microbes. Those stromatolites are like the best bet because those are very easy to see. They're macroscopic, they're easy to see, but it's really hard to find fossilized cells of microbes. One thing that they are looking for are what they call biosignatures, which is certain chemicals that the microbes might produce that are still lingering. And so it's like it's evidence that the microbes were there. So sometimes they'll look for that. When they refer to these ancient cells, by the way, they often use the term protocell. That just means first cell. And so often you'll hear that term. So protocell just means the very, very primitive cells. And when I was taking my first microbiology class in undergrad days here, we were taught that microbes were probably forming, the cellular life was probably forming along the ocean shore. And that has since been changed, where they think maybe not. They're actually saying the subsurface of ocean, and they're actually saying near hydrothermal springs or hydrothermal vents in the ocean floor, more so than the shore of the ocean. And so we'll get into why that seems to be an optimal place for cellular life to come into being. But we gotta wait because I think we ran out of time, didn't we? So we'll stop there. Thank you for your patience, and I'll see you all on Wednesday.